Have you ever looked at your family and wondered why you have your dad's nose but your mom's eyes? Or maybe why your dog has spots, but its puppies have a totally different pattern? These questions are at the very heart of genetics, the amazing science of heredity and variation. Inheritance is the process of passing down traits, or characteristics, from parents to their children. It's the reason why apple trees grow apples and not oranges, and why kittens grow up to be cats, not dogs. It's nature's way of creating a blueprint that gets passed from one generation to the next, a set of instructions for building a living thing. But here's the cool part. That blueprint isn't always copied perfectly, and that's where variation comes in. Variation is simply the set of differences we see among individuals in a group. Think about it. No two people are exactly alike, not even identical twins when you look really, really closely. This variety is what makes life so interesting and so resilient. It's why some people can run faster, why some flowers are brighter, and why some animals are better at hiding from predators. Without variation, every living thing of a certain kind would be a clone, and a single disease or environmental change could wipe them all out. Understanding inheritance and variation is not just a fun thought experiment, it's fundamental to understanding life itself. It explains the beautiful diversity we see in the natural world, from the stripes on a zebra to the different shapes of leaves on a single tree. It helps us understand where we came from and gives us clues about our own health and biology. This science is a powerful tool. It allows us to grow better food, fight diseases, and protect endangered species. So, let's dive in and explore the incredible science that makes each of us a unique, one-of-a-kind masterpiece of nature. Consider this your official invitation to unlock the secrets of life. The journey into genetics is like being a detective, piecing together clues that were left behind by generations before us. Every cell in your body contains a history book written in a special code, and scientists have been working for centuries to learn how to read it. By studying how traits are inherited and why variations occur, we can solve some of life's biggest mysteries. It's a story that starts in a quiet monastery garden and leads all the way to high-tech laboratories that can map out our entire genetic code. It's a story about what connects us all, and at the same time, what makes each of us so special. So, put on your thinking caps because we're about to get to the science of it all. Our story begins with a truly remarkable scientist, a monk named Gregor Mendel, who lived in the 1800s. Mendel was a curious fellow, and he was fascinated by the pea plants in his monastery garden. He noticed that some pea plants were tall, while others were short. Some had purple flowers, while others had white, and some produced smooth peas, while others had wrinkled ones. He wondered if there was a pattern to it all. So, he started doing experiments, carefully crossing different pea plants and recording the results over many generations. You could say he was the original data scientist of genetics, and his work laid the foundation for everything we know today. Mendel discovered some fundamental rules which we now call his laws of inheritance. The first one is the law of segregation. Mendel figured out that for each trait, an organism inherits two factors, one from each parent. We now call these factors genes. He realized that these two factors separate or segregate from each other when reproductive cells, like pollen or egg cells, are made. This means each parent passes on only one of their two factors for a trait. For example, if a tall plant has one tall factor and one short factor, it can pass on either the tall one or the short one, but not both at the same time. Then Mendel came up with his second big idea, the law of independent assortment. This law states that the factors for different traits are sorted into reproductive cells independently of one another. In simpler terms, getting the gene for tallness doesn't affect whether you get the gene for purple flowers. Imagine you're making a sandwich. Choosing to use wheat bread doesn't force you to use turkey. You can still choose ham or cheese. In the same way, the gene for seed color, say, yellow or green, is passed on independently of the gene for seed shape, like smooth or wrinkled. This shuffling of genes creates a ton of possible combinations in the offspring. Mendel's experiments were brilliant because he kept it simple. He chose traits that had only two clear forms, like tall or short, and he counted everything meticulously. For example, when he crossed a purebred tall plant with a purebred short plant, all the offspring were tall. He called the tall trait dominant. But when he crossed these new tall plants with each other, about 1/4 of their offspring were short. The short trait, which he called recessive, had been hiding all along. His careful work showed that inheritance wasn't just a blending of traits but a passing down of distinct, predictable units. It was a revolutionary idea. Mendel's work with dominant and recessive traits was a fantastic start. But as scientists studied more and more organisms, they realized that genetics can be a bit more complicated. Sometimes one gene isn't completely dominant over another. This is called incomplete dominance. In this case, the organism with two different genes for a trait shows a phenotype that is a blend or an intermediate between the two. It's like mixing paint. If you mix red paint and white paint, you don't get red or white, you get pink. The same thing can happen with genes. A classic example of incomplete dominance is seen in snapdragon flowers. If you cross a purebred red snapdragon plant with two red genes with a purebred white snapdragon plant with two white genes, you might expect to get all red flowers if red was dominant. But that's not what happens. Instead, all of the offspring will have pink flowers. The red gene isn't strong enough to completely mask the white gene, so they blend together to create a brand new in-between color. This shows that inheritance isn't always an either-or situation. Sometimes it's a beautiful compromise between what each parent contributes. Now, let's talk about another cool variation called codominance. The prefix co means together, and that's exactly what happens here. Both genes are expressed together at the same time, but they don't blend. Instead of making a new intermediate form like pink, both of the original traits show up. Imagine taking that red paint and white paint, and instead of mixing them, you paint red and white stripes. You can clearly see both original colors. A great example of codominance is found in certain types of chickens, like the erminette chicken, which has both black and white feathers, creating a speckled or checkered look. Another very important example of codominance is in human blood types. The A and B blood types are codominant. If you inherit an A gene from one parent and a B gene from the other, you don't get some blended blood type. Instead, you have AB blood type, where your red blood cells have markers for both A and B on their surface. Both genes are fully and equally expressed. These examples of incomplete dominance and codominance show us that the world of genetics is full of fascinating complexities, and that Mendel's simple rules are just the beginning of a much bigger, more intricate story of life's code. So, we know that genes are passed down from parents to children, but why is there so much variety? If we're all just inheriting instructions, why aren't we all more alike? The answer is that the genetic instruction book is constantly being edited, shuffled, and added to. One of the main sources of this genetic variation is mutation. A mutation is simply a change in the DNA sequence, a tiny typo in the genetic code. These changes can happen randomly when a cell is dividing and copying its DNA. Most mutations have no effect, and some can be harmful. But every now and then, a mutation creates a new trait that can be beneficial. Think of a mutation as a random change to a recipe. Most of the time, adding a random ingredient might not change the taste, or it might ruin the dish. But once in a while, you might accidentally add cinnamon instead of nutmeg and discover an amazing new flavor. That's what a beneficial mutation does. It creates a new version of a gene, called an allele, that didn't exist before. This is the ultimate source of all new genetic traits, from the first blue eyes in humans to antibiotic resistance in bacteria. Without mutation, evolution would grind to a halt, because there would be no new material for natural selection to work with. Another huge source of variation is genetic recombination, which happens during sexual reproduction. This is like shuffling a deck of cards. Before parents pass on their genes, the chromosomes inside their cells get together and swap pieces. This process, called crossing over, mixes up the genes from the parent's own two parents, the offspring's grandparents. So, the chromosome you get from your mom isn't an exact copy of her mom's chromosome or her dad's chromosome. It's a unique mosaic of both. This shuffling creates brand new combinations of genes on each chromosome, ensuring that siblings, except identical twins, are genetically different from each other and from their parents. Finally, we have gene flow. This is what happens when individuals move from one population to another and reproduce. When they do, they bring their genes with them, introducing new alleles into the new population. Think of a group of birds with only brown feathers living on one island. If a few birds with a gene for blue feathers fly over from another island and join the group, their blue feather gene is now part of the local gene pool. This increases the genetic variation of the population on that island. Together, mutation, recombination, and gene flow are the powerful forces that constantly stir the genetic pot, creating the incredible diversity of life we see all around us. Nurture. It's not just about your genes. While your genes provide the fundamental blueprint for who you are, they don't tell the whole story. The environment you live in plays a huge role in shaping your traits. This is, the classic nature versus nurture debate. Nature refers to your genetics, and nurture refers to your environment. Everything from the food you eat, to the air you breathe, to the experiences you have. The truth is, it's not a competition, it's a partnership. Your genes and your environment are constantly interacting with each other to produce your final phenotype, or your observable characteristics. Consider the hydrangea plant. The color of its flowers is not determined solely by its genes. Instead, the acidity of the soil it's planted in makes all the difference. In acidic soil, the flowers will be a beautiful blue, but in alkaline, or basic soil, the very same plant will produce pink flowers. The plant has the genes to produce colored flowers, but the environment flips the switch that determines which color is expressed. This is a perfect visual example of how an external factor can directly influence how a gene works. The same principle applies to us. For example, a person might have genes that give them the potential to be tall, but if they don't get proper nutrition during childhood, they may not reach their full potential height. In recent years, scientists have discovered an exciting new layer to this interaction called epigenetics. The prefix epi means above or on top of, so epigenetics literally means above the genetics. It refers to chemical tags or markers that attach to our DNA and can turn genes on or off. These epigenetic markers don't change the DNA sequence itself, but they change how the DNA is read and used by the cell. Think of it like this: your DNA is the cookbook and the genes are the individual recipes. Epigenetic markers are like sticky notes placed on the recipes, saying, make this one often or skip this one for now. What's really fascinating is that your environment and lifestyle choices can influence these epigenetic tags. Things like diet, stress, and exposure to certain chemicals can add or remove these sticky notes, changing which genes are active. Even more incredible, some of these epigenetic changes can even be passed down from one generation to the next. This means that the experiences of your parents or even grandparents could have left a subtle mark on your gene expression. This field is, honestly, revolutionizing our understanding of health and disease, showing us that the connection between our lives and our biology is even deeper and more dynamic than we ever imagined. Gregor Mendel was a genius, but he was really working in the dark. He didn't know what his factors were made of, or how they actually worked inside the cell. It took almost a century for science to catch up, and finally discover the physical basis of his laws, DNA. We now know that Mendel's factors are actually genes, which are specific segments of a long, complex molecule called deoxyribonucleic acid, or DNA. This DNA is tightly coiled into structures called chromosomes, which are found in the nucleus of our cells. The discovery of the DNA double helix in 1953 unlocked the molecular secrets of inheritance and connected Mendel's abstract rules to a real, tangible, physical reality. This new molecular understanding allowed us to see that inheritance is often way more complex than the simple dominant or recessive patterns Mendel saw in his piece. For example, many traits aren't controlled by just one gene. They're influenced by multiple genes working together, a phenomenon called polygenic inheritance. Poly means many, so it's literally many genes. Traits like human height, skin color, eye color, and weight are not either-or traits. They exist on a continuous spectrum, with lots of shades and sizes in between. That's because they're the result of the combined effects of many different genes, plus environmental influences. Another layer of complexity is sex-linked inheritance. In humans and lots of other animals, sex is determined by a special pair of chromosomes, X and Y. Females usually have two X chromosomes, while males have one X and one Y chromosome. Some genes are located on these sex chromosomes, which means they're inherited differently in males and females. For example, the genes for red-green color blindness and hemophilia are located on the X chromosome. Because males have only one X chromosome, if they inherit a recessive allele for one of these traits, they will express it. Females, however, have a second X chromosome that can carry a dominant, healthy allele to mask the recessive one. So, while Mendel's laws gave us the perfect starting point, modern genetics has built upon his work, revealing a much richer and more intricate picture of heredity. We now understand that genes can interact in complex ways, that some traits are linked to an individual's sex, and that the environment plays a crucial role. We can read the DNA sequence itself, identify specific genes responsible for diseases, and even trace human ancestry back thousands of years. We've moved from counting peas in a garden to mapping the entire human genome, but the fundamental principles of inheritance that Mendel first uncovered still hold true. Understanding inheritance and variation isn't just for scientists in labs. It actually has a huge impact on our everyday lives. One of the most important areas is medicine. By understanding the genetic basis of diseases, we can develop better ways to diagnose, treat, and even prevent them. For example, we know that certain genes can increase a person's risk for conditions like heart disease, diabetes, and some types of cancer. Genetic testing can help people understand their personal risk, allowing them to make proactive lifestyle changes or undergo more frequent screenings. This is really the foundation of personalized medicine, where treatments are tailored to an individual's unique genetic makeup. And think about agriculture and the food we eat. For thousands of years, humans have been using the principles of inheritance without even knowing the science behind it. They saved seeds from the best plants, the ones that were biggest, tastiest, or most resistant to drought, to plant the next year. This is called selective breeding. Today, our understanding of genetics allows us to do this with much greater precision. Scientists can identify the specific genes responsible for desirable traits, like higher crop yield, better nutritional value, or resistance to pests. This technology helps us feed a growing global population and make farming more sustainable. The science of genetics is also a critical tool for conservation biology. When a species is endangered, its population becomes small, and it often loses a lot of its genetic variation. This makes the species more vulnerable to disease and changes in the environment. Conservation geneticists can analyze the DNA of animals in the wild to measure their genetic diversity. This information helps them make smart decisions about how to protect and manage these populations. For example, by designing breeding programs that maximize genetic variation, or by creating wildlife corridors that allow isolated populations to connect and share their genes. From solving crimes with DNA fingerprinting to tracing your family tree through ancestry tests, the applications of genetics are all around us. It helps us understand who we are, where we came from, and how our bodies work. It gives us powerful tools to improve our health, secure our food supply, and protect the planet's biodiversity. The study of inheritance, which started with simple observations in a garden, has grown into a science that is fundamentally changing our world for the better and empowering us to tackle some of humanity's biggest challenges. It's science in action, and it's happening right now. With great knowledge comes great responsibility. As our ability to understand and even manipulate DNA grows, we're faced with some very important ethical questions. It's one thing to understand the gene for a disease. It's another thing entirely to change it. Technologies like CRISPR allow scientists to edit genes with incredible precision, almost like a find and replace function for DNA. This has the potential to cure devastating genetic disorders like cystic fibrosis or Huntington's disease. But it also opens up a conversation about where we should draw the line. Should we use this technology only to cure diseases, or could it be used to enhance humans? One of the biggest debates revolves around the difference between somatic and germline gene editing. Somatic editing targets the body cells of a single person, and the changes are not passed on to their children. It's a form of personal therapy. Germline editing, on the other hand, would change the DNA in reproductive cells, sperm or eggs, or in an embryo. This means the changes would be permanent and would be inherited by all future generations. While this could potentially eradicate a genetic disease from a family line forever, it also means we would be altering the human gene pool. The long-term consequences of such changes are unknown, and many people feel this crosses a significant ethical boundary. There are also serious concerns about privacy and fairness. Your genetic information is the most personal data you have. Who should have access to it? Should insurance companies or employers be able to use your genetic information to make decisions about your coverage or job prospects? There's a risk that this could lead to a new form of discrimination, where people are judged not by their actions, but by the predispositions written in their DNA. We need to create strong laws and ethical guidelines to protect genetic information and ensure that everyone is treated fairly, regardless of what their genes say. These are not easy questions, and there are no simple answers. They require careful thought and public discussion involving scientists, ethicists, policymakers, and all of us. As we continue to unlock the secrets of our own biology, we must proceed with caution, wisdom, and a deep respect for the complexity of life. The power of genetics is incredible, but how we choose to use that power will define our future. It's a global conversation, and everyone's voice is needed to help us navigate this new and exciting frontier responsibly and for the good of all humankind. From Gregor Mendel's humble pea plants to the cutting-edge labs that map entire genomes, our journey to understand inheritance and variation has been nothing short of extraordinary. We've learned that inheritance is the beautiful, intricate process by which life passes its instruction manual from one generation to the next. It's a set of rules, first sketched out by Mendel, that explains why we resemble our parents. It's the reason life has continuity, a thread connecting the past, present, and future. This blueprint ensures that species remain recognizable over time, carrying forward the traits that have allowed them to survive. At the same time, we've discovered that this instruction manual is not set in stone. Variation is the essential ingredient that adds spice, surprise, and resilience to the story of life. Through mutations, the shuffling of genes during reproduction, and the influence of the environment, life is constantly experimenting and creating new possibilities. This diversity is not just beautiful to look at, it is the single most important factor that allows life to adapt and thrive in an ever-changing world. It is the engine of evolution, the raw material from which nature selects the best solutions to the challenges of survival. The principles of inheritance and variation are more than just a chapter in a biology textbook. They are fundamental to who we are and the world we live in. They shape our health, our food, and our environment. Understanding them gives us the power to heal diseases, feed our population, and protect the incredible biodiversity of our planet. This knowledge has equipped us with tools that our ancestors could only dream of, placing us at a unique point in history where we can actively and intentionally shape the future of life on Earth. As we move forward, it is crucial that we embrace this knowledge with both excitement and humility. The story of our DNA is the ultimate story of connection, linking us to our families, to our ancestors, and to every other living thing on the planet. By continuing to explore the elegant dance between inheritance and variation, we not only uncover the secrets of what makes us who we are, but we also learn how to be better stewards of the amazing, diverse, and precious phenomenon we call life. The blueprint is in our hands, and the next chapter is ours to write.